Good evening, everybody, and thank you for joining us for Cafe Sci. My name is Dan Lavoie, and I'm honored to be with you as the host this evening. Cafe Sci is presented by PPG. Cafe Sci is also supported by the good people like you. Thank you to everyone who donated to support the program. If you are unable to join and not be able to donate, but would still like to be able to support Cafe Sci, the most helpful thing you can do is invite your friends to join the audience. Cafe Sci takes place the first Monday of each month, and you can register for next month's speaker at carnegiesciencecenter.org. Before I introduce tonight's speaker, I'd like to remind everyone that you can ask questions by using the Q&A button at the bottom of our Zoom window. You can enter your question at any time during the presentation. All right. It's my pleasure tonight to introduce Dr. Rachel Hostetler. She's a scientist here at Allen Institute for Brain Science in Seattle, Washington, where her role is to increase the accessibility of the Allen Institute's open data sets to researchers, educators, and the public. She supports research through workshops, tutorials, seminars, and more. With the public, she has participated in talks, hands-on activities, and podcasts to introduce the importance of open science and the kind of basic research done at the Allen Institute. Prior to working at the Institute, she completed a PhD in neuroscience at West Virginia University, where she studied the diversity and connectivity of different neurotypes in the cerebral cortex. Welcome.
Thank you.
So I'm going to turn it over to you. Again, thank you for being here. We're very excited to dig in and learn about this exciting topic. So without further ado, I'm going to give you the floor.
Perfect. Sounds great. And thank you for the introduction and thank you for inviting me here today to present this exciting work. All right. So, as mentioned, I'm Rachel Hostetler, and today I'll be talking about brain cell atlases or mapping the uncharted territory of the brain. And a brief background about me. I've been involved in neuroscience research for the past 15 years. I initially did my bachelor's of science at the University of Minnesota, where I double majored in neuroscience and German. Then, as mentioned, I got my PhD in neuroscience at West Virginia University, where I did a lot of imaging and other types of neuroscience experiments to study different types of neurons in the cerebral cortex. And then now here at the Allen Institute for Brain Science, I've been here for about 2 years now. And my position, as mentioned in the intro, really deals with helping and supporting technical users of our data sets and resources, running workshops, running seminars, and also doing some public talks. And what is the Allen Institute? So we're on the other side of the country over here in Seattle, Washington. And the Allen Institute is a nonprofit research institute that was founded by Paul Allen and Jody Allen back in 2003. And Paul Allen was one of the co-founders of Microsoft. And the Allen Institute has grown since its initial founding. It started with about 4 employees, and now we're up to over 900. And being a nonprofit research institute, the goal is to really focus on understanding the basic principles of research and of science, and then using that data to advance research all around the world. And much of the data I'll be talking about today is considered basic science or basic research. And if you haven't heard of basic research before, it's sort of the beginning part of the research pipeline. So we have basic research, we have translational research, and then we have clinical research. And many people are aware of clinical research when they hear about clinical trials going on to test new drugs or new treatment. But there's a lot of research, sometimes decades of research that happens before those clinical trials can even start. And so basic research is really looking at the beginning. What types of cells do we have? What's going on in different organisms? If there's a disease that we're studying, how does that disease affect certain cells? It's really looking at what's happening at that basic foundational level. And then in between, we have something called translational research, where we've studied the cells, we've studied the disease, we have an understanding of how the disease works, but now we'd like to potentially come up with treatments that could target that disease or treat those cells. And so again, the majority of this talk will be focused on basic research, that really foundational research. And so here at the Allen Institute, we're guided by a couple core principles. The core principles being big science, meaning we do really large research experiments that require hundreds of people working together. team science, where we actually have individual teams doing certain parts of the experimental pipeline, which differs a little bit from academic research or academic science, where typically you have one lab doing many steps of a process. Here we have different teams doing different steps, which I'll explain a little bit in a few more slides. We also believe in open science. So all of our data, our research, our protocols is all available freely online for anyone to access and use. And we also believe in high impact research. So large projects that really make a global impact on the neuroscience community. And going a little bit into open science. So again, I mentioned we have our data available and fully public for anyone to look at. In addition to publishing our data, we also publish our protocols. So if there are scientists in other labs around the world interested in learning more about our methods, they can go online and search for our specific protocols and look and learn how we do things exactly step by step. We also provide resources and trainings so that others can help use and understand our data and tools. And we also believe strongly in FAIR principles, which means making sure that data is findable, accessible, interoperable, and reusable. And here are some advancements from the Institute over the last 20 years or so. So I mentioned that the Institute was founded in 2003 by Paul Allen and his sister Jody Allen. And one of the very first projects that the Institute created was the Mouse Brain Atlas, which can be seen here in the bottom corner. And so in neuroscience, mice are used for a lot of research. We are very similar to them. Brain-wise, we have a lot of similar neurons. We have similar gene expression patterns in our brains compared to mice. So mice are really used foundationally for a lot of different neuroscience research. And so this Mouse Brain Atlas was released in 2006, and it was used and has been used and is still used by neuroscientists all over the world today, even in 2025. Since then, we also released a Human Brain Atlas, a Connectivity Atlas, which looks at the different connections between neurons within the brain, a Cell Types Database, an Alzheimer's Brain Cell Atlas, which I'll talk about a little later in this talk, an Immune System Explorer, and also an Allen Brain Cell Atlas. And so all of these different atlases come from a couple different institutions. So I mentioned the first institute is the Institute for Brain Science. This is the institute that I work in. And then over time, over the last 2 decades, other institutes have been formed within the Allen Institute. So we now have the Allen Institute for Cell Science as well, the Allen Institute for Immunology, and also for Neural Dynamics, which is a second neuroscience institute. But the majority of this talk today will be about research that is from the Allen Institute for Brain Science and is focusing on foundational neuroscience research. And so earlier I mentioned team science. So what exactly does team science mean? So there's about 900 people who work at the Institute, and they're divided into different teams that interact with different parts of the experimental pipeline. So a typical scientist in a research lab might have to do every single one of these steps by themselves, or their team might have to do all of these steps. That's usually how things are a bit more structured in academic scientific environments. But at the Allen Institute, we actually have separate teams of people specifically devoted to each of these steps. And so this makes every step of our research pipeline very standardized because it's the same people every single time working on each individual step. And this also helps us produce very large data sets that would not be able to be produced in smaller research labs. And so these are different people and different teams involved in the experimental pipeline. But we also have a number of other important people as well. Those involved in project management, those are involved in research compliance, finance, shipping and receiving. It's a very large operation to do these very large scientific experiments. And so all of the work that I'll be presenting today is work that has been produced by hundreds of people working together in a really collaborative environment. So what exactly will I be talking about today? The brain, that's the focus of today's talk. Initially, We'll talk a little bit about, what we know about the brain and the different brain areas. So, over the last 200 years or so of neuroscience research, we know some pretty standard things about the brain. We know its typical size and shape, the different parts. We know that there's different lobes in the brain and that different parts of the brain have different functions. And a lot of this we've known for, over 100 years or so, because, back in the 1800s, there were a number of accidents that happened, where someone gets an injury in one part of the brain, and then you notice they have certain symptoms or they have certain functions that they lost. So a lot of early neuroscience research was based on mapping out the large areas of the brain based on injury or illness that happened in different brain areas. And so because of that, we know that if you have an injury to your frontal lobe, sometimes people have changes in personality. If you have a stroke, um, also in that area near your motor cortex, you usually have some type of motor or movement deficit. If something happens to the cerebellum in the back of your brain, you can have difficulties with balance and coordination. And so these are the very large, gross areas of the brain that we know about. But can we dive deeper into the brain? We know we have these big areas, but what's going on in the brain? There's billions and billions of neurons in individual cells within the brain. And so our goal at the Allen Institute is to figure out at the cellular level, what are these billions and billions of cells doing in the brain? And this is a question that neuroscientists have been asking for over a century as well. So initially back in the late 1800s, there was a famous neuroscientist. He's usually called the father of modern neuroscience. His name was Santiago Ramon y Cajal. And he did these immaculate drawings of neurons, where he would look at neurons under a microscope and trace them by hand. And that was a really, really important thing at the time when he was doing this. This was the first time that we could really see that neurons within the brain are individual structures and that they connect to each other to form these larger circuits. And he could also notice that not every neuron was identical. They had different shapes, they had different projections coming out of them. And so for a long time, neurons were categorized simply just by their shape. So sometimes they got kind of funny names. Like someone named a neuron the chandelier cell because it's sort of spread out like a chandelier. And in 2025, chandelier cell is still used today as a name for that type of neuron. Some of them were named pyramidal because they were sort of shaped like a pyramid. Like for instance, this cell down here, cell number E, this would be a typical pyramidal cell because its cell body here is a bit pyramid shaped. Some of them looked like baskets, so they were named basket cells because they kind of enveloped other neurons. And so again, we knew long ago that neurons had different shapes. We didn't really know why they had different shapes or what if these different shapes were important, but we knew that they had different shapes and names were given to them based on their shapes. Later in the mid 1900s or so, 1940s, 1950s, 1960s, a lot of experiments were done to understand and to realize that neurons actually have electricity inside of them. So neurons send messages through each other and they have electrical signals. And that's what's shown here in this figure here. These are different kinds of neurons and they have different electrical signals that they send. And so this was another step. We could figure out neurons' shape, and they were named based on their shapes. But now people started naming them based on their electrical patterns, which were called firing patterns. And so some of them had really fast firing patterns, and so they were given the name fast spiking. Some of them had maybe faster and then slower, so they were called adapting. Some of them were just called irregular spiking. These are just names that neuroscientists have been using for over half a century now to just identify cells, neurons in the brain based on their electrical patterns. Then a couple decades later, the late 1900s or so, there were more experiments done and new ways to categorize brain cells as well. And so with new techniques, we could learn that some neurons had certain molecules inside of them that were not in other neurons. So they were named molecular markers. And so a technique called immunohistochemistry was used to identify and label these different molecular markers in individual neurons. And so here, what we could learn was that, these types of neurons, that's what these white blobs here, these are individual neurons, and they're glowing in this image because using the technique immunohistochemistry, we could see that they had a certain peptide inside of them that these other neurons did not have. So these are different kinds of neuropeptides that are present in different types of neurons. And we could learn that not every neuron has the same molecules inside of it. So these molecules are in there for some reason and they have some sort of function. And so we've been spending a long time figuring out what exactly are these molecules doing. But they were used as a way to sort of identify and label different markers of neurons. So those were the main previous techniques that have been used to sort of categorize and catalog different cells in the brain, but there are some limitations. You could only measure a small number of cells at the same time. So the human brain has tens of billions of cells. And with all these techniques, you could maybe only get, if you're recording their electrical activity, you have to do that usually one cell at a time. That takes a very long time. You can't do that for billions of cells. When you use these methods, you could only get a small number of features from each cell. And what do I mean by that? So if you're looking at the cell's electrical activity, that's all you're looking at. There's so many other properties of that cell, like maybe its shape or maybe something to do with its genes that you're not aware of because you're not measuring all those other properties. You're just measuring one thing. And many of these techniques are very time consuming and involve a lot of manual labor from neuroscientists in the lab running these experiments. But up until around 2010 or so, these were sort of the main ways to identify different brain cells in the brain. And around that time, a new technique was invented that allowed neuroscientists to measure thousands of features from thousands of cells much faster. And so that technique is called single cell RNA sequencing. And you might see this abbreviated later in this talk as scRNA-seq. But the point of single cell RNA sequencing is that it gave us a new way to look at these different brain cells. And so going back to the central dogma, we have DNA. DNA gets transcribed into RNA, which then gets translated into proteins. And when we're doing single cell RNA sequencing, we're focusing on the RNA here, the transcriptome, which means we're interested in studying all the different RNA transcripts in each cell. And so here again, pretend this is a cell here. We have our DNA. It gets transcribed into mRNA. And then with single cell RNA sequencing, this is a technique where we can actually measure the number of RNA transcripts for each gene in an individual cell. So you're measuring maybe 30,000 different genes in one individual cell. And then you can do that with thousands and thousands and thousands of cells all at the same time. And when you do that for a very large number of cells, that's an incredible amount of information. That's many, many gigabytes of information, potentially more. But when you get all this information from thousands of cells, what you can do is you can start to organize cells based on their similarity to each other. So if you've thought about the animal kingdom, right, the animal kingdom is based on different classes and different divisions. And, humans were in the mammalian group, and then we're in the primate group, right? You have all these different groups nested within other groups. And we have a similar type of organization here in the brain. We have some cells that are genetically very similar to other types of cells, which are located within a larger family of cells which are then very distantly related to maybe some other type of brain cell over here. And so just like mapping out, the animal kingdom and how similar we are to different animals, this is what we're trying to do in the mammalian brain. Map out all the individual cells, all the neurons, see how similar they are to each other, and map out how they connect to each other in a larger hierarchy. And so this here is also called a taxonomy. And so you might hear that word later in this talk as well, talking about different taxonomies of cells within the brain. And so, as I mentioned before, this technique, single cell RNA sequencing, it's pretty new. It was first published in 2009, and the first study involved looking at genes from only 4 individual cells. And that was groundbreaking at the time. That was extremely important. That was brand new. And then over the years, there has been new developments in technology and in analysis that has allowed more and more data to be collected throughout the experiments. So this graph here ends in 2017, so it's a little out of date, but even at 2017, we were getting around to measuring 1 million cells all at the same time. So looking at tens of thousands of genes in a million cells and then comparing those million cells to each other to see how similar or dissimilar they are to each other. And so this technology has really caused an explosion of data, not only in the neuroscience world. This technique is also used for cancer research. It's used, in other organs. It's used in, research for the lung or for the heart. But in brain research, it's really also taken off and, caused a really big explosion in data. And so if we go around here to 2016, what we can see is most of these studies were around maybe 1,000 or so cells. And so in 2016, a study came out from the Allen Institute, again, looking at the mouse brain, and they were able to sample and catalog about 1,600 different cells. And then using that RNA data from each individual cell, they could compare them to all the other cells and build out this nice diagram here where we can see these 2 cells in the bottom right corner, are much more similar to each other genetically than, the cell is to the one all the way over here, right? So every time we sort of have a split here in this diagram is where we have a divergence between different kinds of cells. And cells that are grouped similar to each other in our diagrams are much more genetically similar to each other than to cells from farther away in that diagram. And this was specifically looking at the area of the brain locate in the back of the head in the visual cortex for processing visual information. And so again, these cells were collected, analyzed, and from all this genetic information, we could see that there were about 49 different cells that could be identified within this experiment. Jump forward to 2023. Again, we've had many advancements in technology and analysis. All of a sudden, now we're able to do millions of cells at the same time and have a lot more data and a lot more different types of cells in the brain. So there's been a huge advancement in the last, 6, 7, 8 years, which has given us way more data than we had before and way more information about each individual brain cell. And so this study was published in 2023, and 8 million cells were involved in this study. And when they categorized all these different cells and looked at their expression, they could see that there were about 5,000 different kinds of cells in the brain. And so what you're looking at here, this big messy thing, this is a plot. And within this plot, there are 4 million cells. If you zoom in on here, you actually see individual little dots. And each dot is representative of one cell that was used in this experiment. And right now, all the dots in this plot are color-coded based on those 5,000 different cell types. So all of the yellow ones up here are close together, all of these purple ones down here are close together. And these are all quite different than the cells on the other side of the plot, which are very different colors. So if you've heard of principal component analysis or a PCA plot, this is a similar type of math that is used to really just reduce very high dimensional data into 2 dimensions. This particular plot is called a UMAP, and it's just a way to show very high dimensional data in 2D. But the main point is that dots that are closer to each other in space in this plot are much more genetically similar to each other than dots that are far away from each other on different sides of the plot. And so there were a couple different types of experiments that were done in this project, and another one involved actually mapping out each of these little dots in the actual mouse brain here. So this is what a mouse brain looks like. Um, it's smooth on the top, unlike the human brain. The human brain is much more wrinkly, but we have pretty much all the same parts as the mouse brain. So this is a section here, and then here are the different sections throughout the brain. If you were to go anterior to posterior, you can actually map out the 4 million different cells here and where all those different little dots are. And when you have over 5,000 different types of cells, it gets much harder to make one of those really big, taxonomy trees here. So here we actually have it split, but we don't go all the way down to the 5,000 level because it would be a bit too big. So this was the largest data collection of cell types in the mouse brain to exist in the world when this was published back in 2023. So this was an extremely large data set. It made the news. People were very excited about it. It was a really big data release that is being used by neuroscientists all over the world. And you might wonder, well, that's great for mice, but we're not mice. What about, human research? And, I'm here to tell you that we also are doing experiments, very similar experiments, but on the human brain as well. Back in 2023, there was also a very large human atlas that was produced by the Allen Institute. And it also got picked up by the New York Times and by the Wall Street Journal and a few other media outlets. And one of the reasons why mice were focused on first is because, the mouse brain is very tiny compared to the human brain. So first, if you're going to perfect a technique or write a new protocol, it's better to start on something smaller, master it in that smaller sample, and then scale up to the human brain. All right, so I mentioned in 2023, we also had this human brain cell atlas, which was released. This is a very similar type of plot to the one I showed earlier, meaning each of these colors actually represent different types of cells that were identified in the brain. And there's about 3 million different dots here, and all the dots are clustered together, and dots that are really close to each other are similar, and dots that are really far away are dissimilar. And so this was the first draft that we have of a whole human brain cellular atlas. And this was also very exciting when it came out. I showed you that it was in the news. This was very groundbreaking for the neuroscience community. And it really is a first draft. There are current experiments going on right now at the Institute and with their collaborators to improve upon this project and make, a second version of it that's new and improved. And so here's one of those dendrogram, one of these little trees here again, showing at the bottom here, we have all these different kinds of neuron types. And we can actually, using the RNA information from each of these cells, actually see how they're similar to each other and how they're dissimilar to each other. And these atlases have really been a very large collaborative effort. So a lot of the work that I just talked about previously has been funded through the Brain Initiative Cell Census Network, which is through the NIH or National Institutes of Health. And these projects are not just at the Allen Institute. These very large projects involve researchers at institutions all over, specializing different types of technologies. And here's just a plot here showing that many people are involved in these projects. And these projects have been continuing Most of the data I showed you so far was from the last couple of years, but there are more projects underway, and we're actually entering a new phase of this Brain Initiative project here, as you can see from this headline. So there's a lot of work that has been done already, and I showed you some of those published atlases, but there's a lot more coming down the pipeline that you'll see in the next year or so. And so why are we making these brain cell atlases? So far I've shown you maybe some confusing plots, but what can we learn from this? Why is this important? Well, first off, we don't know all the different types of cells we have in the brain. We don't know that information. We know that there's about 180 or so billion neurons and support cells in the brain, but what we don't know is what are all those different brain cells doing normally in a healthy brain. We don't know how they change with aging, and we don't know how they change with disease. We don't know that right now at the cellular level. This is all still new information for us. And so there have been a few studies at the Allen Institute trying to see if and how these brain cells change over time. And so there was another study that came out just this year, actually, in mouse. And this one also got picked up by the news, figuring out how do certain brain cells change over age. Because someone might think, well, maybe all of your brain cells change with age. But it turns out that's not true. There's only a couple smaller groups that either had their genes go up or go down in certain changes throughout age. So it's not a global thing that you see happening. And that's new information for us. We didn't know that before, what exactly is happening at the cellular level. And so here's one of those big plots again. Again, we have millions of cells here. They're mapped out in two-dimensional space. Those colors represent different kinds of cells, and each color consists of many, many dots, which represent individual cells. And if they're closer together, they're more similar. If they're far away, they're less similar. And so this was looking at all of the cells together in this experiment. And then in this plot here on the right, here we're just looking at particular cells that were affected by age. And so in green here, these particular cells actually had genes that were depleted with age. And over here, this brownish color, these represent cells that actually had their genes enriched with age. So it's not like you have, one group of cells that's all affected in one way. We actually had some going up and some going down. So that's also new information. And what exactly are these cells here? So these ones down here in green that had genes depleted, these are actually some of our oligodendrocyte cells, which are a support cell in the brain. Some of these produce myelin. Perhaps you've heard of myelin. It creates a sheath around the neuron and allows it to send electrical signals. And there are certain diseases like multiple sclerosis where that myelin is affected. And then over here in this brownish color, some of these cells that had their genes enriched are microglia. And microglia are immune cells that are active in the brain. And in general, that study found a lot of different exciting things. Here's a figure from the paper just sort of summarizing all of that. So with age, again, this is a study from mouse. So we don't know if this will exactly translate to human, but at least from this study in mouse, we could see that there were some changes in the immune response in the brain. There was an increase in inflammation, and that there was a decrease in some other functions. There was a decrease, and there was an effect happening to our oligodendrocytes here. Some of our neurons lost some of their structural integrity. There's a decrease in neuronal signaling. And there are also some changes that are potentially both increase and decrease with our homeostasis, with certain neurons involved in energy levels. And so we were able to do this research and do these experiments because we had the original baseline atlas to compare the data to. So we'd already had a brain atlas for the mouse, and now we could compare the cells from the older mice to that original atlas as a baseline and see how are things changing with aging. What else can we learn from these atlases? Well, we can also learn how the brain changes with disease. So over the last few years, the Institute has been working on a very large Alzheimer's cellular atlas, which was reported on by NPR. And from this very large data set, again, here's one of those confusing plots. Colors mean similar types of cells. And if they're clustered together, it means they're similar. If they're far away from each other, it means they're dissimilar. And here were all of the cells together in one group. And then the researchers, because they were looking at Alzheimer's, they had a number of samples um, from postmortem samples from people who had passed away, who had volunteered to be part of a study involved in studying Alzheimer's disease. And so some people who volunteered for the study had very high levels of Alzheimer's, and they had very high dementia, and they had a lot of pathology in their brain. And some people in this study had very low levels of pathology. They had no dementia. And so because of that, there was a variety of disease severity in this particular study. The researchers could actually map out the progression of Alzheimer's disease and not just look at the really severe version. And so they could map out over time what's happening at the cellular level when you look at beginning, early phase of Alzheimer's versus very late phase of Alzheimer's. And so some interesting parts of this study here is, and this plot on the right, it's the same thing as here on the left, but instead I'm just showing cells that were related to Alzheimer's disease. So most of these cells here, most of these dots are again microglia, which are those immune cells. And also some oligodendrocytes, again, which are those support cells in the brain that support the neurons and create a myelin sheath. And so what was interesting about the study was that they genetically, when looking at the RNA, they could see that there were microglia in the Alzheimer's brains that were new. They were somehow genetically different than all of the microglia from the non-Alzheimer's brains. So there's some sort of shift happening that's causing these changes. And because they were mapping out the brain cell changes over time, here's a figure from the paper that sort of lays that out earlier. In these particular figures here on the left means early Alzheimer's, so before you even have any actual plaques or tangles in the brain. And then on the right here is when you have more severe Alzheimer's. And so what they learned was there were certain types of neurons here, these are neurons, that died very, very early in disease, way before you even have the presence of plaques and tangles. And then there were other types of neurons that were lost later in disease. Right here, you see this decrease here, this green line. And so that's new information. We didn't know that before, that different types of neurons within the brain are affected differently by Alzheimer's disease, and that they die off at different times throughout the progression of disease. So this is new information. And we were able to do this study because there was a previous study before looking at healthy human brains. And so because of that, they were able to compare the Alzheimer's brains to the healthy brains and see where do we have these differences? Where do we differ from our healthy baseline? And as I mentioned previously, there were not just changes in the neurons. You have a ton of cells in your brain that are not neurons. You have a lot of support cells, which are called glial cells, which encompasses these oligodendrocyte cells that I've mentioned a few times, and your microglia, those immune cells. And so throughout the progression of disease, you actually have this increase in microglia in the brain. You have more inflammation. And you have a loss in some of these oligodendrocyte cells down here. And so now there's more research being done on this particular project because we don't know exactly why these particular neurons are lost so early in Alzheimer's disease. And, another question is if somehow you could prevent these cells from dying, with a medication or a therapeutic, does that stop the rest of the disease from progressing forward? We don't know. That's sort of the next step is figuring out, okay, now that we know that different brain cells are affected at different stages of disease, can therapeutics be developed to interfere there or, see what happens if you stop that progression? So that's exciting. And, those are some new directions for this particular study. And as I mentioned, a lot of the work that I presented here today is done by hundreds of people. And this particular project is a very large consortium where we partner with the University of Washington and Kaiser Permanente Washington and the Alzheimer's Disease Research Center. And this is a project that is funded through the National Institutes of Health, through the National Institute on Aging. And there's also a website where you can learn more about this project as well. All right, so most of the data I've talked about so far was really just looking at RNA in individual brain cells and then using RNA to compare brain cells to other brain cells and looking at their similarities and differences. But if you remember at the beginning of the talk, I mentioned that neurons have different electrical activities and they have different shapes. And we now know that they also have different gene expression or they have different RNA in their cells. And so another technique has been developed recently, also within the last 10 or so years, that combines all of these three modalities together. And that technique is actually called patch-seq. And so this is really interesting because if you're just doing these types of experiments, you're just looking at the RNA of each individual cell, you're missing a lot of information. You're missing all of the information about its function, so its electrical signaling, and also its connectivity. So neurons connect to other neurons and form these giant webs, but not every neuron connects to every other neuron. There's a very selective process, and they form these very specific circuits, and we're still discovering and still researching what these circuits are. And so this technique allows you to look at different types of information and sort of integrate it together. So here's another big one of those plots. Again, this is looking at different types of cells in the brain of mouse, particularly in the motor cortex, so the area for movement. And on the right-hand side here, we have all of our non-neuron cells. So I mentioned, we have these immune cells in the brain. You have a lot of endothelial cells, so cells for your blood vessels. We have a lot of other support cells, those oligodendrocytes I've mentioned a couple of times. So those guys are all over here because they have very different RNA compared to all the neurons way over here, right? And if you look at this chart, you can see which ones are more similar and which ones are more different. And so this is just looking at, again, the RNA from the individual cells. But if you use the technique patch-seq that I mentioned, you can do many types of experiments at once. So you can also look at their morphology. And what you can see here is that these neurons really have very different shapes. Some are much more elongated and much more spread out, and some are much more condensed. And this is really important because this determines the other cells that they interact with. So a neuron that's really condensed and has really short arms makes synapses with other neurons that are nearby, whereas neurons with really long arms that reach all the way across the brain make synapses with neurons that are much further away. So they're involved in very different circuits in the brain. So the shape is very important. And then in addition to shape, they have different functions and different behaviors. Remember, the neurons have that electrical activity, and some of them have very fast electrical activity where they have very fast rates of sending their electrical signals. Some are much more slower. And so this technique allows us to look at different types of modalities of the cells all together. And this technique is much more difficult, so you can only do it on, really one cell at a time. So if you end up with a collection of thousands of cells like this, it's quite impressive because there was a lot of manual labor that went into doing all of those experiments. But that's another way to map out cells in the brain. I also have one more example. So another way to map out all the connections in the brain is by using a different technique called electron microscopy. And so electron microscopes have been around for quite a long time. They're very, very powerful microscopes that allow you to see things at a much greater resolution than a regular light microscope. You can see things at 100,000 times better resolution with an electron microscope because electrons are so much smaller than photons of light. And so, because of this, if you want to look at a synapse, so synapse is when two neurons are able to connect, there's a tiny space between those two neurons, and that's called the synaptic cleft. And it's impossible to see the synaptic cleft with a regular microscope that uses light because it's so small. So you have to use an electron microscope, which uses an electron beam to do the imaging to be able to actually see that synaptic cleft between the two different neurons. And so a project has been going on at the Allen Institute in collaboration with the Baylor College of Medicine and Princeton University to map out all of the neurons in all of their synapses within 1 cubic millimeter of mouse brain. And a cubic millimeter is very, very tiny. It's about the size of a grain of sand. But this project resulted in petabytes of data. It took about 7 years, and they mapped out 200,000 cells and about 500,000,000,000 different cellular connections. And this paper, there were about 10 papers that were published with this project a few months ago, back in April. And I really like this description here. Scientists map part of a mouse's brain that's so complex it looks like a galaxy. And that's true. You can go in and look at this data set yourself. It's free online for anyone to look at. I'll show you the website in a few more slides. But this is an unprecedented level of resolution of looking at all of the cells in a very, very small part of brain in all of their connections. And here's a video just showing that. So in this video here, there's one neuron in the center. It's that blue color. It's a pyramidal neuron because it's sort of shaped like a pyramid. There's a green cell wrapped around it. That green cell is actually a microglia. It's one of those immune cells. And then all of those tiny purple dots along the neuron are all of the synapses that our other neurons are making onto that neuron. So there are thousands and thousands of connections being made onto this one individual cell, and that's what all of the purple dots represent. So this is truly what neurons look like in the brain, and you can only see it at this level of detail and resolution with an electron microscope. And again, this particular neuron comes from a data set in the mouse brain. So again, why are we mapping out all these different brain cell types? Why do we need these atlases? It's cool to see how brain cells change, but what are some other advantages? Well, it turns out once you've mapped out and cataloged different cells in the brain, then you can start to develop tools to target those specific brain cell types, specifically for gene therapy, right? So if you're going to give someone a gene therapy, you don't want it to affect, 50 billion different neurons in the brain. If only one type of neuron is sick, you want to develop a tool, a gene therapy that only targets that particular type of neuron. And so first off, you need to know what kind of neuron is sick in this disease. Is it just one type of neuron? Maybe it's two different types of neurons. What are their characteristics? What's going on with those neurons? And then because we know that these particular groups are vulnerable in disease, how do we develop tools to then target them and just them and not the other neighboring neurons? So that's really a new avenue that's happening at the Allen Institute and also at other research institutes around the world. This very large data set was just released also this year in 2025. This is from the Washington Post. I like how they describe these genetic tools as gene delivery trucks, because that's sort of what they are. They're little delivery trucks that just go to certain particular cells and deliver whatever they need to deliver. And so this is really at the beginning, developing these tools. And then hopefully in the future, maybe some of these could become actual gene therapies in the future. And this is also funded through a different project through the National Institutes of Health. And meanwhile, at the Allen Institute, we have a team that's also been working on a separate gene therapy project, specifically looking at a seizure disorder called Dravet syndrome. And Dravet syndrome is a very severe seizure disorder. And through a lot of the research at Allen Institute, they've been able to identify which exact neuron is having problems, which neuron is malfunctioning in this disorder, and then can we correct it in a mouse using a gene therapy? So that's some really active research that's going on at the Institute right now. That's very exciting. And some other ways to use these data sets as well. So I mentioned all these things that we're doing at the Institute, but other researchers around the world also use these data sets. So here's a paper that I found a couple weeks ago. It was a research paper written by scientists in St. Louis, and they used the data from our brain atlases and said, oh, we're going to look through these atlases and we're going to find new things and write new papers about the new things we're discovering. A number of educators also use these data sets for teaching in high school and at the undergraduate level, and also at the graduate level. So here is a paper here from a teaching professor at UC San Diego, where she was using a lot of these data sets to teach her undergraduate neuroscience students how to do programming, how to do coding. So because we want this data out there, it's open for anyone to use. You don't need permission to use it. People use it in a variety of ways for both research, for education, and also for other types of outreach. And any of you can also access these data sets. So a lot of the data I showed in the beginning, that RNA sequencing data, can be found at brain-map.org. All of the electron microscopy data, so those really, really detailed renderings of neurons, can be found at microns-explorer.org. And if you're interested in learning more about the Alzheimer's data set, that's at sea-ad.org. So it's all free, it's all open for anyone to use for whatever they would like. In addition to putting our data sets online, some of these data sets are quite complex. We've also created, our technology team has also created a number of tools to help people use these data sets. So here's an example of an interactive tool. You don't need to download anything. You can just look at it in your browser. But this tool is called the Allen Brain Cell Atlas, or ABC Atlas. And I showed some of these plots earlier. These are the different cells that were analyzed in that big mouse brain project that I mentioned. And here you can actually go through and click on these plots. You can learn about the different types of neurons and different types of cells that exist in the brain. And you can also search for different genes. So I searched a gene that I like because I'm curious, where is this gene expressed in the mouse brain? So that's also an option that you can do here. Just search through here and find this different type of information. And this type of resource is really, really beneficial for other neuroscientists, researchers around the world. Prior to working at the Institute, I was doing my PhD in neuroscience, and I heavily relied on, well, this data set didn't exist yet, but the previous versions of these data sets I used for my own research to help guide my own experimental questions, because it was such large data sets that I personally could not have produced. So I relied on other people's data, to help support my findings. And so that's really, really important in the research field, in science. You really want to compare your results to previous people's results and see how you match up or how you differ. And this is a great way to do that because these data sets are so large. All right, so here are some key takeaways from the talk. I know I showed a lot of different kinds of data and a lot of different kinds of plots, but if the plots and data were confusing, I want these things to be understood. Science is often self-correcting. So I showed in the very beginning, I showed that, researchers 100 years ago were using different techniques to study the brain compared to researchers today. And that doesn't mean that the researchers 100 years ago were wrong. They were correct in their understanding of the brain at that time with the tools that they had. But now we've had such an advancement with tools and techniques that now we have a new understanding of the brain. And again, it doesn't mean that the previous people were wrong. We've just gained new understanding because we have new data, because we have new tools to use. Science is also very, very collaborative. So all the data that I showed, hundreds and hundreds of people from multiple institutes and universities contributed to that work. When you have really large projects, working together, you have different areas of expertise. It's really helpful and science moves forward when you have all these big groups working together. And open science allows us to advance research faster. So I mentioned during my PhD work that I looked at a lot of these data sets or similar data sets to help guide my own research questions. And that saved me a lot of time because I didn't need to repeat a bunch of experiments. I could look at what someone else did, what hundreds of people did, rely on that data and say, okay, that makes sense based on my own research. And now I can move forward and plan my other experiments. And basic research, which is mainly what we talked about here today, really lays the foundations for tomorrow's treatments and cures. So if you want to, create a new treatment for a certain disease, first you need to know which cells in the brain are sick in that particular disease, which means you need to study how the cells act when they're healthy in the brain, right? You need to have that baseline understanding to understand the diseased brain, and then also to be able to develop any cures or treatments. All right, and thank you everyone for this talk. We do want to thank our founder, Paul Allen, for his support, and also the NIH grant that supported this work as well.
Thank you very much. That was absolutely fascinating. And we do have some questions from our audience, some from different portions of it. So I apologize if we're going to ask a question that may have been answered later on, but it's still great. And we can't wait to dig into these. So first, are there really 5,322 different cell types, or are some different stages of the same cell?
Yeah, so that is a question that a debate that sort of goes on with neuroscientists and cellular biologists a lot, the difference between a cell type and a cell state. Yeah, that is a good question. I think it's still up for debate, but I think in this particular study, the way that they were defining using the transcriptomic data, they were able to definitively say that these are actual cell types. And especially looking at the brain, the cell states are, yeah, you don't have as much change in your cell states in the brain as well. But I also will say that this work is still in progress. So there's another mouse brain atlas that has been produced by another group. And then the next step is comparing, our mouse brain atlas to their mouse brain atlas and seeing where do things diverge, where are we similar. So I don't necessarily think that, yeah, 5,322 is the final number, but that's the number that we have currently with this data set.
Fantastic. Next question is from Nancy Truvers. So the human brain has fewer types of cell types than mice.
Yeah, so if you were to look at those two different data sets, that's something that you would think. Because there's an order of magnitude more of just neurons in the human brain compared to the mouse brain. And I will say that human brain atlas is really considered a first version draft by the lead researchers of that project, and they're currently working on another one. And in that particular human brain atlas, there were a few areas that were slightly undersampled compared to other areas, and that can deal with just the techniques that you use to do these experiments. Some areas of the brain are more resilient and survive the experimental process, and some are less resilient. So I think, yeah, I'm gonna assume there would be more cell types in the human brain, but we don't have the actual number yet. But those are the numbers that we have currently.
All right, now the next question is interesting, and I want to take that question and kind of spin it off into a little bit of a larger one. The question from one of our attendees was, assuming you work on brains from people who are deceased, how do you keep the brain properly active to do your research? And I kind of want to dig in a little bit that more and talk about what you do get out of doing research with deceased brains.
Yeah, so there's a couple of different experiments that go on in the Institute that work with human tissue. So all of the work from the Alzheimer's data set, that is postmortem tissue. So those brain samples end up being frozen and stored and processed that way. But there are a number of experiments done at the Institute with the technique that I mentioned called patch-seq. And if you're recording the electrical activity from cells, the cells need to be alive. So you cannot do that with frozen tissue. And so in those cases, the Institute partners with local hospitals, and we're actually able to get brain samples from people who are undergoing some type of brain surgery, usually for sometimes seizures or cancer research. And the neurosurgeon will then remove a healthy part of the tissue, call someone at the Institute, and then bring that sample over to the Institute while that tissue is technically still alive and you can still record the electrical activity in it. So those projects are very, very time sensitive, right? Because there's a lot of moving parts. Um, there's actually a really interesting news article on our website, alleninstitute.org, where they interviewed someone who had brain surgery and he donated part of his brain, and just his experience going through that, and how he felt, contributing part of himself to science, because it's a very selfless decision to do that.
That is fascinating and interesting. I wasn't sure the depth of which you would be able to utilize a deceased brain. It sounds like there is quite a bit of things to learn from someone who does choose to donate their brain in this kind of situation.
Yes, in the post-mortem brains, I will say that there are some brains at the brain bank that have been there for, someone maybe donated their brain a decade ago or 2 decades ago. If they're stored properly, there's a particular way that they're stored that's still extremely valuable information and can be used for research, even today.
That is absolutely interesting. So next question is from Ned Uber. Have people applied these tools to the simple animals where the whole connectome is understood?
Yeah, so a lot of so if people have heard about Drosophila or fruit flies. Drosophila is a research animal, the little tiny fruit fly that is used for a lot of neuroscience research as well. So their entire connectome has also been mapped out. So yeah, it's true that I only mentioned examples from mouse and humans, but there are other smaller things. I mean, a lot of the things we now know in mouse were really originally studied in those tiny little fruit flies. A lot of genetics work. Um, that applies to humans. A lot of foundational genetics work has been done in much smaller organisms.
This is an interesting question next from Robin Oliverio. So I apologize. I probably butchered your last name. Do you know what the exclusion criteria for the non-AD human atlas were? For instance, for a lot of clinical studies, patients with a history of substance use disorders are excluded.
Yes, so I don't remember off the top of my head what the exclusion criteria was, but it is in the main paper that I mentioned. Um, but it is correct that especially Alzheimer's also does overlap with a lot of other disorders, not only substance use disorders, but also neurodegenerative disorders. So if you were to just eliminate everything, eventually you'd have a sample size of 0. So that's another thing that's you have to be aware of when dealing with human samples, especially because human samples are so precious. That is someone who selflessly donated themselves, right? You don't want to just eliminate everything. So substance use disorder, I don't remember exactly, but I could get back to you with that information. But it is available on our website. I know it's on there listed.
Another question here. I apologize if I butchered this question. Is there more research being done in Parkinsonism, and what is the new help available? A little bit of a
Yes,
summary.
we just had a new data set released actually about a few weeks ago that I did not mention in my slides. That is actually a collaboration that we've been doing with samples from Parkinson's disease, partnering with the Michael J. Fox Foundation. And it's on our website. I guess if you were to search, I guess, Allen Institute Parkinson's, we had a few news articles written about it. That should pop up. But that's also a brand new study that was just released, and that is looking at comparing those Parkinson's samples to the Alzheimer's samples that we have and also to the healthy brain samples. Because there's also a lot of overlap between, like I mentioned, different neurodegenerative disorders and perhaps are the same types of cells that are sick in Alzheimer's, are they also sick in Parkinson's, or is it completely different? Those are questions that we can now start to ask because we've actually integrated those data sets together.
All right, we have one more question, and this one is a little bit in a different direction. So I apologize if it depends. It's totally okay if we want to take it in a different direction, whatever you'd like. Is there any thought to the brain is more of an old-fashioned analog computer rather than the digital computer that we all know? Any articles on that?
Yeah, I'm trying to think of any particular articles on that. I
Yeah,
can't think of any off the top of my head, but it is, we're still mapping out exactly what, if one neuron is getting like 1,000 inputs and another, the other neighboring neuron's getting also 1,000 inputs, and then, it's hard to realistically think about all those different inputs coming together. It's not just a one-to-one connection between all these different cell types. So I think as we increase in complexity, we'll have to, it's a little overwhelming to think about all the different
right.
ways that it's all connected.
Right. Well, that is all the questions we have tonight. I just wanted to thank you for coming out and being our speaker and giving us a really fascinating topic. I know we had about 40 or so folks in here enjoying this live, and you'll be able to catch this on YouTube on our Carnegie Science Center's YouTube page here soon. So I wanted to thank you for being here and thank all of our wonderful guests for being here to listen tonight, and have a really great evening.
All right. Yes. Thank you for having me.
Bye, Rachel. Have an excellent evening.